Hello and welcome to my channel. In this lecture, we will conclude our talk about the epithelium. Let's start. We described, we talked about the types of epithelium and the locations of the epithelial tissue and the functions of the epithelial tissue. And now we'll go into little details about the junctions between the cells of the epithelium to make it, ready to perform its function. So the topic will be cell junctions. Cell junctions are present in the epithelial tissue, and they consist of various protein complexes that hold the cells together and ensures a contact between a cell and the neighboring cells and the extracellular matrix. And this is essential also to control the transport across the cells, across the basement membrane of the cells. And there are five major types of cell junctions: the tight junctions, the adherence junctions, the desmosomes, the hemidesmosomes, and gap junctions. Tight junctions, they are junctions present at the near the apex of the apical side of the cells, as you can see here, holding the apices together. And it's composed of a transmembrane protein fused on the outer plasma membrane. And I want you to take a look at this. This is an example of a tight junction. See the protein complexes holding the plasma membranes together. Different types of protein present there. You don't have to learn them by heart, but know that it's responsible for formation of the junction itself, and also stress on the fact that it's near the apex. These are the tight junctions. Adherence junctions, it's represented here in this picture. I want you to pay attention to this protein, which is called cadherin. Cadherin you will find in many types of junction. It's the protein that holds the two cells together. And also pay attention to what it holds, what structures inside the cell it holds together. In this case, it holds the micro or the actin filaments, the skeleton of the cells together, of two adjacent cells together, as you can see. Each cadherin molecule has heads that hooks around the actin filaments, and tails that goes into the bilayer of the plasma membrane, holding the two cells together. We will stress on the cadherin molecule, and in this type of junctions we'll stress on the microfilaments. So it holds the microfilaments together, which are actin filaments. Another protein responsible for this type of junction is called vinculin. It's associated with adherence junctions in addition to several other actin binding proteins, and it's responsible for this type of junctions. So this is an idea about the composition of the adherence junctions. A desmosome, the one represented here in this picture. Now also cadherin is present there, as you can see in the picture, but what it holds in this case is not actin filaments but keratin molecules. It's also known as macula adherens. It's specialized for cell-to-cell adhesions, and it's a type that localized spot-like adhesions randomly arranged on the lateral sides of plasma membranes, on the lateral sides, not only at the apex, as mentioned before, in tight junctions. Desmosomes attach to microfilaments of cytoskeleton made up of keratin protein, not actin but keratin protein. So in this case, cadherin is also there, but it holds keratin instead of the actin of the two cells together. It's the same principle, but it's what it holds that is different. A hemidesmosome, it resembles a desmosome in a way, but instead of cadherin molecule, they use integrin molecule, a transmembrane protein, instead of cadherin. And they attach the epithelial cells to the basement membrane. So this is a type of junction that attaches the cells to the basement membrane. So now you studied different locations of junctions. The apical junctions, the lateral side junctions, and the junctions that attached the epithelial cell to the basement membrane. Gap junctions, they ensure a free passage of ions between the cytoplasm of two adjacent cells. So it's a connection between the two cytoplasms by a protein called connexins. Each connexin, there are 6 connexins joined together to form a connexon. Each connexon goes through the plasma membrane to contact the other or the adjacent cytoplasm, and as you can see here, it can be closed and open to allow for free passage of ions for rapid spread of impulses coming to the epithelial or rapid exchange between the epithelial cells. Functions of the epithelium: to protect the tissues that lie beneath, especially of course if it's stratified, to protect it from radiation or dryness, from toxins, invasion by pathogens, and physical trauma. Also, it regulates the exchange of chemicals between the underlying tissues and the body cavity. It's responsible for secretion of hormones into the circulatory system, and also the secretion of mucus and enzymes and sweat, and substances that are delivered by duct by the exocrine glands. Also, it provides sensation, as mentioned. The sensory or the olfactory mucosa is a type of epithelium, modified neuroepithelium. The hair cells in the inner ear are other type of cells that are responsible for sensation, the sound sensation. It also absorbs water and digested food in the lining of the digestive canal. These are the major functions of epithelial tissue, or the different functions of the epithelial tissue according to the location they are present in. Glandular tissue The glands, exocrine glands, are formed by infoldings from the surface epithelium and subsequent growth of the underlying connective tissue. You can classify glands into endocrine glands and exocrine glands. Endocrine glands, they secrete their secretion directly into the blood, into the circulatory system. They don't have a duct, and it's rapidly taken in this way. But exocrine glands, they secrete into duct system of the gland, and the duct system opens into the lumen of an organ or onto the free surface of the epithelium, like the sweat glands, for example, the exocrine component of the pancreas, for example. They have a duct system. You can classify the glands according to their shape into simple alveolar or simple acinar. This is only present during development. Simple tubular, a single tube invaginating from the surface epithelium, present in the intestinal glands. Simple coiled, they are the sweat glands. Simple coiled tubular, and simple branched tubular, that tube that branches off, and this what you can see in gastric glands. Branched alveolar, branched acini, the example of which is sebaceous glands. Now, there are compound glands, means that they have multiple of these varieties, multiple acini or multiple tubules and acini. You have compound alveolar or acinar, example of which the mammary gland. You see multiple acini developing from the single invagination. Compound tubulo-alveolar, multiple tubule and acini from the invagination of the surface epithelium, and compound tubular from branching of many tubules from a single invagination. The compound tubulo-alveolar examples are salivary glands and glands of the respiratory passages that secrete mucus, and the pancreas. The compound tubular examples are mucus glands in the mouth, the bulbourethral glands in the male reproductive system, and testis in the seminiferous tubules. So this is the classification of glands according to their shape, according to their mode of secretion into exocrine and endocrine. According to their shape, it's the classification you're looking at in this slide. Also for the mode of secretion, for example, we have merocrine secretion, example of which are sweat glands. They simply form the substance to be secreted into vesicles, and the vesicles are expelled out by means of exocytosis, as you can see here. The Golgi complex is responsible for packaging of the secretion and forming of the vesicles to be excreted. Another form of excretion present in the mammary gland called the apocrine secretion. Apocrine secretion means that a portion of the cell, secretory cell, containing the secretion is pinched off. A portion of the cell is pinched off. This is what happens in mammary glands. Another mode of secretion is holocrine secretion, present in sebaceous glands. Now, for this gland to secrete its contents, it must die first. So it dies and the plasma membrane ruptures and the excretions inside are expelled to the outside after the gland dies. So we have merocrine secretion, apocrine secretion, and holocrine secretion. This is the classification of glands according to the mode of secretion. One of the functions of the epithelium is sensing of the extracellular environment. This is a picture of the pseudo-stratified columnar ciliated epithelium. These are the cilia of the respiratory tract with the electron microscopy of the respiratory epithelium. These cilia have chemosensation and thermoreception and mechanoreception of the extracellular environment, and so they play a sensory role, and as a consequence they have a secretory role in which they secrete mucus to expel foreign materials to the outside of the body. This is an example of the sensing of the extracellular environment by the respiratory epithelium. Also, it has a role in immune response because it has can express many genes that encode immune mediators that attract the components of the immune system to a site of injury, for example. And this is encoded in the genome of these cells, which enables a rapid response to immunological challenges that encounters the body. Now, for a hint about the clinical significance of epithelium, it's a common site for cancers. Originating our epithelium are called carcinomas. Well, you can apply the word sarcomas for cancers that develop in the connective tissue. In case of cystic fibrosis disease, in which there's I think abnormal absorption or utilization of chloride, sweat glands are also damaged, causing a frosty coating of the skin. This is an example of affection of epithelial cells. I hope you got the maximum benefit from this lecture, and thank you very much for watching.